In the next two lectures, we'll discuss degradation of feed. We'll begin today with polysaccharides. By the end of these two lectures, you'll be able to explain steps of feed degradation, including attachment, hydrolysis, and fermentation. You'll be able to explain steps of hydrolysis specifically, including the enzyme systems involved, and describe the structure of substrates and how it can limit hydrolysis. Your reading will be Chapter 12 from the textbook. We'll talk about degradation of feed by handling each of the substrates within feed separately. In this lecture, we'll talk about polysaccharides in the form of the cell wall and in starch. In the next lecture, we'll talk about protein. There are multiple steps of degradation: attachment and ingestion, hydrolysis, and fermentation. We'll begin by discussing attachment and ingestion. As we've mentioned before, about 3/4 of bacteria are attached to feed particles, and we can see that in the figure to the right. The plant particle is covered with bacteria. One of the reasons for attachment is that soluble nutrients are scarce except after feeding. The most reliable source of substrate is in the feed particles. There are several stages in attachment to feed particles for bacteria. First, in Part A, we have association of primary colonizers. By definition, these are the bacteria that arrive to the feed particle first. In Step B, we see the primary colonizers adhering to the feed particle with binding proteins. This ensures close and continued attachment to the feed particle. Relatively few primary colonizers will attach to the feed particle. They proliferate and cover the feed particle by dividing, which you see in C. In D, we see the association of secondary colonizers. This is a second group of bacteria that comes into close contact with the feed particle after the primary colonizers begin to degrade substrate. They are not firmly attached as the primary colonizers, just closely associated. Once substrate is depleted from the feed particle, bacteria will disassociate, as we see in E. After a time, as we see in F, feed particles will pass from the rumen, and any bacteria that are attached or associated will pass as well. The primary colonizers for the cell wall are cellulytics like Fibrobacter succinogenes, Ruminococcus flavifaciens, and Ruminococcus albus. For the endosperm, which contains starch particles, the primary colonizers are the amylolytics, such as Ruminobacter haemophilus. The secondary colonizers are cross-feeders. They're feeding off some of the degradation products released by the primary colonizers. We'll talk more about cross-feeding later in this lecture and the next one. Ruminococcus albus requires phenylpropionate and phenylacetate for attachment. In an earlier lecture, we explained that cellulytics require phenolic acids as a growth factor, and this is what explains that requirement. Phenylpropionate and phenylacetate are required specifically because they stimulate the formation of the cellulose-binding proteins of the cellulosome. We'll reference these cellulose-binding proteins and the cellulosome later, but for now, just know that these cellulose-binding proteins are the ones that affect attachment of the bacteria to the cell wall. You can see the effect of withholding phenylpropionic acid on Ruminococcus albus in the image to the right. At top, we have healthy Ruminococcus albus that's been given phenylpropionate, and you can see a healthy glycocalyx surrounding the bacteria. They can attach to feed particles. At bottom, we have Ruminococcus albus that has not been given phenylpropionate. It does not form a very thick glycocalyx, and it cannot bind to feed particles. Fungi also attach to feed particles. They do so when zoospores embed themselves in particles and form thalli. You can see thalli attached to a plant fragment in the SEM image to the right. Both Entadyniomorphid and isotrichid protozoa attach too. Further, the Entadyniomorphids ingest small fragments that have been broken off of the particles. The isotrichids can ingest starch granules only. After microbes have either attached or ingested feed particles, they begin hydrolysis. During hydrolysis, polysaccharides are converted to oligosaccharides and then die in monosaccharides. and you can see that to your right, where the polysaccharides include cellulose, hemicellulose, and pectin, which are part of the cell wall, and starch, which is found in the endosperm. This hydrolysis is not an easy task, given the complex structure of the cell wall and the endosperm, which contains starch granules. We'll discuss the structure of each of these in subsequent slides. To begin with the cell wall structure, we see the cell wall is a matrix of cellulose, hemicellulose, pectin, and lignin. Protein is a more minor component. The figure at right shows the structure of the cell wall, including the middle lamella and primary layers. Cellulose is in a dark green. Individual chains of cellulose form bundles, which are the microfibrils we see in the figure. These microfibrils run on top of each other. We see hemicellulose in a slightly lighter shade of green. This runs between and cross-links the cellulose microfibrils, providing strength to the cell wall. In the primary wall, pectin is in orange color. It also runs between and cross-links the cellulose microfibrils. In the middle lamella, which is the space that runs between adjacent cells, pectin is a light green. Here, it serves as a glue that cements adjacent cells together. Lignin is absent from this figure because it's not part of the middle lamella or the primary layer. Lignin is a part of the secondary wall, as we can see in the figure to the right. It is covalently bonded to hemicellulose and possibly also cellulose. The arrangement of lignin and cellulose in the secondary cell wall provides strength and rigidity to the cell. It does this in a similar way as steel rods embedded in concrete, where the steel rods are the cellulose microfibrils and the lignin is the concrete. Because of the complexity of the cell wall, its hydrolysis requires cooperation. The figure at bottom shows all the microbial species involved and their interactions during hydrolysis. We'll go over each of these steps in detail. First, the cellulytics, the primary colonizers, degrade the cellulose in the cell wall. As they degrade the cell wall, they release oligosaccharides originating from cellulose and hemicellulose. You can see this action in the current figure, with Ruminococcus and Fibrobacter, the primary colonizers, releasing solubilized oligosaccharides and polysaccharides. The cross-feeders, the secondary colonizers, consume the oligosaccharides being released. These include the Prevotella species, Butyrvibrio fibrosolvens, Selenomonas ruminantium, and others. You can see this process highlighted in the figure. The Butyrvibrio and Prevotella species are taking up the solubilized oligosaccharides and polysaccharides. Roseburia and Bacteroides are also shown, as we discussed their species in the human colon, and they serve a role analogous to Butyrvibrio and Prevotella. There are two enzyme systems involved in hydrolysis. One is the cellulosome, which is cell-bound, and the second system consists of free enzymes. The cellulosome is a complex of multiple proteins that work synergistically in cellulose hydrolysis. We can see a figure of the cellulosome to the right. To discuss these proteins and the figure in detail, we see first catalytic subunits. Those are the units in the figure that look almost like Pac-Men. They have hydrolytic activity towards the cellulose. They're cellulosomal enzymes, and they're the ones that are affecting the hydrolysis. There are also structural proteins in the cellulosome. These are scaffoldins. As the name implies, these form a scaffolding. This scaffolding accomplishes two things. First, it holds cellulosomal enzymes, and second, it binds cellulose. The subunits that hold cellulosomal enzymes, SCA, are shown in yellow, green, blue, and purple. The subunits that bind cellulose are cellulose-binding proteins. This is CTTA, and it's shown in a light blue. As we mentioned earlier, phenylpropionate and phenylacetate stimulate the formation of the cellulose-binding proteins. The cellulosome is not the only way that cellulose can be hydrolyzed. There are cellulases that are free. These are cellulases that are not bound to the cell, but rather released into the milieu. The cells still have cellulose-binding proteins, but the cellulases are not attached. Ruminococcus flavifaciens has a cellulosome, as we saw in the preceding figure. Ruminococcus albus probably also has a cellulosome. Fibrobacter succinogenes, by contrast, has free cellulases. Thus, we see both of these enzyme systems with the cellulytics. Microbial cells have evolved complex machinery to hydrolyze cellulose, but there are still limits to hydrolysis. The factor that controls the rate of hydrolysis is surface area. Untreated cellulose is fermented very slowly, only between 5 to 8% per hour. However, if we finely grind the cellulose, it's fermented very quickly. The experiment shown in the figure at the right shows fermentation of very finely ground cellulose or cellobiose by Fibrobacter succinogenes. The formation of fermentation acids from cellulose was very rapid. It was fermented at 68% per hour initially. This is just as fast as cellobiose, which has the same linkages as cellulose, but it's a disaccharide. There is no surface area limitation for its hydrolysis. The factor that controls the extent of hydrolysis is lignin. It is strongly related to the indigestibility of NDF. The figure at the right shows indigestible NDF after a 96-hour in situ incubation, and we see that it's very well related to the lignin in NDF. The mechanism by which lignin limits hydrolysis is physical. Lignin itself cannot be degraded, and it physically shields the cell wall from hydrolytic enzymes. We'll continue our discussion by talking about starch hydrolysis, beginning with an overview of the structure of the endosperm that contains the starch granules. The endosperm is seed tissue that contains starch granules in grains. Starch is not found in free form. It consists of a protein matrix which surrounds the starch granule. We can see the structure of the endosperm in the figure to the right. In panel A, we have starch granules present with the protein matrix behind them. In panel D, we have the starch granules alone. There is no protein matrix. To hydrolyze starch, we first have to hydrolyze the protein matrix in the endosperm. This is the slow and rate-limiting step in starch hydrolysis. Once the protein matrix is hydrolyzed, the starch granule itself is hydrolyzed very quickly. The figure at the right shows the endosperm of corn after being incubated with strep bovis. You can see that the starch granules are completely absent, but the protein matrix is intact. This is consistent with the idea that the protein matrix is slowly hydrolyzed, and only once it's hydrolyzed can the starch granules be quickly hydrolyzed themselves. What you see here should explain why many amylytic bacteria are also proteolytic. Just like for cellulose hydrolysis, there are cell-associated and free enzyme systems for starch hydrolysis. For the cell-associated system, starch is bound extracellularly and then hydrolyzed to oligosaccharides. The oligosaccharides are transported into the periplasm, hydrolyzed, and then transported into the cytoplasm. The figure at right shows the system for Bacteroides theta iota omicron, which is a bacterium from the human colon, as you recall. But the system for Rhinobacter ammophilus seems similar. The same genes are present, and biochemical experiments show a similar process. These cell-associated systems cause concentrated pitting in starch granules. You can see this in a starch granule of wheat after incubation with strep bovis, with one of the pits pointed out to you. This is a good way to visualize the action of this system. Free amylases are also present. They're not associated with the cell, but rather released into the milieu. Most bacteria seem to have both systems. Our last step of degradation is fermentation. We'll very briefly cover it today, because a future set of lectures on energy metabolism will discuss fermentation pathways in great detail. During fermentation, glucose is converted first to pyruvate. Pyruvate is then subsequently converted to the VFA, acetate, butyrate, and propionate. It's also converted to lactate, CO2, hydrogen, and methane. Fermentation yields ATP, which is used to fuel microbial growth. And as I mentioned, we'll talk much more about this later. The take-home messages are that polysaccharide degradation requires multiple steps, including attachment and ingestion, hydrolysis, and fermentation. The complex structure of the cell wall and the endosperm explains the need for the involvement of so many species and the development of complex enzyme systems. Both cell-bound and free enzyme systems contribute to hydrolysis.